Right. All right. In that same vein, Mr. Saddle. All right. In that same vein, what are these? What? What? No, I heard something else.
Does it start with an S?
No, I thought I heard diatoms. Did I hear diatoms? Huh? You said diatoms, right? Somebody did? Diatoms. Say it louder. Those are diatoms. Okay? Those are diatoms. That's all you need. Diatoms. Okay? Nothing else. All right? Those are easy. So we have diatoms and we have dinoflagellates. All right? All right. So let's see. Uh-oh. Huh. I'm missing a couple of things. I don't like that. Darn it. All right.
I hope my phone can hold the 30-minute video. I tried to delete as much as I could.
I'm doing voice recording pictures.
So, dadgummit. Who is messing with these? None of these are in order. What do they do with this? Ah, here we go. They probably do shake them up. I don't doubt it. All right. So, who is this? Volvox. Very good. It is Volvox. What do I call the little tiny dots? The little tiny itsy dots. Those are individual cells. What do I call the little smaller balls inside of that? Those are daughter colonies. Okay, daughter colonies. The name of that guy is Volvox. What kind of algae is it?
Red. No, green.
It's green algae. It's stained red. So no. It's actually a green algae. Okay, so it's actually a green algae, and it's is it filamentous? Is it unicellular? Is it colonial?
It's colonial.
Colonial. Very good. Okay, it is colonial. The balls were daughter colonies. The individual little teensy dots were individual cells. All right.
I think of the big balls as a bunch of dots clumped together.
Yeah. All right. So who do we have here? Nothing right now.
Something totally.
Ooh, yeah. That's what all right. Who is this?
Spirogyra.
Very good. This is Spirogyra. What do I call this little prominent guy right here in the center? Nucleus. What are these little dots? Chloroplast. What do I call that? Cell wall. All right, I come over here. What do I call this thing that is joining these two guys? Conjugation tube. So again, what kind of algae is this? Red, green, or brown? Green. All right. Unicellular, filamentous, or colonial? Filamentous. All right, over here, as we move over into the series, I can now determine and distinguish my filaments. Who is this filament? Positive filament. If you put female, it would be fine. Who is this? Minus or male would be fine. All right. And when I get over here, what is this thing? A zygote. Any questions? All right. Also, do make sure that you know your different algae groups. Okay, I don't have any of the gross specimens of algae, but remember, if you're green algae, what phylum are you in? Chlorophyta. If you are in red algae, you are? Phaeophyta. Okay, and if you are, no, did I say brown or red?
You said red.
Who did I say?
Red and green.
Brown algae is Phaeophyta. Who's red algae? Rhodophyta. Okay, so I'm sorry. Chlorophyta is green, Phaeophyta is brown, Rhodophyta is red, okay? And again, you should be able to tell by the color of the algae. I mean, it's pretty obvious. Red algae is red, brown algae is brown, green algae is green, okay? All right, so then let's see. Who do I want to look at? All right, oh, no particular order. So I am just finding them and doing them here. All right. So do some of the simpler ones here really quickly. All right, so what is this thing right here? That is a pollen grain. What would I call that long structure? That would be a pollen tube. Very good. So that's my pollen tube. Okay. Let us see. I'm just seeing a whole bunch of slides over here. All right, let's move over here. And then here we go. All right. So kind of, sort of in the same vein here, keeping with the angiosperms. What would I call this whole thing? The whole thing is an ovary. I am divided up into these little things that are called ovules. How many ovules do you see?
6.
I see 6 ovules. That makes this plant a monocot. Groups of 3, monocot, okay? So if this is my ovule, what do I call this little guy right here? Embryo sac. What would be inside of my embryo sac? There would be an egg, and there would also be, very good, polar nuclei. And if one sperm fertilized the egg, I would get a zygote. If I had a sperm that fertilized my 2 polar nuclei, I would have endosperm. Very good. Okay. All right, so don't forget your incumbas, okay? Don't forget all of your incumbas. We'll look at a few of them as we look at the stems, but remember, go back and review your banana, your pear. Your banana had the parenchyma that had the starch grains in it, correct? Okay? You had the pear, which had your parenchyma, the big cells, and had your sclerenchyma, those little stone cells that you saw there in the pear. And then don't forget your bell pepper. We saw parenchyma that contained the pigments, correct? Okay. And we also found your onion epidermis, right? And we looked at the elodea that actually had the green chloroplast inside, correct? So don't forget that, okay? So then we looked at this guy. Where's my long one? Is this the one I want? Yes, this is the one I want. Not a fan of that one. You can see the xylem really well. Okay, so yeah, it's not as colored as I would like to see. But what is this tissue that I'm looking at right here? Oops.
Vascular.
What kind of vascular tissue am I looking at? Did I hear it? I heard phloem somewhere. Phloem. So this is phloem. These are my sieve tube members. You can see that is that porous part that actually makes it the sieve tube members, because remember that's actually got lots of tiny little holes in there. That's my phloem, correct? What does phloem transport?
Sugars.
Phloem transport sugars.
Isn't that the cucurbita?
That is actually squash. It is cucurbita, yes. All right. So then I have these little guys. So what tissue am I looking at? I am looking at xylem. Okay. Okay, so I'm looking at my xylem here.
So you just stripped
Now, what are my little tiny cells that I see that are very long and narrow and tapered at the ends? Those are just my fiber cells, okay? Those are fiber cells. Then back to that one. Look at a different slide on that one here in a second. But I'm gonna look at this one real quick. Oh yeah, these are much better. I'll look at these and then I'll go back and find the other guys on a different one. All right, so who are all of these that you see right there with all of the big openings? The big openings are my tracheids with the pits. They look like flutes. Big openings. Those are my tracheids with my pits. What I didn't find were vessel elements that I liked. So that's why I switched. So let me see if I can find that a little bit. Those are tracheids.
I could just barely see if this.
With the pits. Let's see. Let me see if I can find some better ones I like. Oh yeah, much better right here. So the ones that tend to have more of the wider structure and the cross hatches, okay, these are my vessel elements. Okay, I'm trying to find a little bit better. Some of them aren't very dark. Oh, right there. Those are some really good vessel elements right there. Where you can really see those cross hatches. They have lots of them. Those are vessel elements, okay? Those are vessel elements. Wider cross hatches. Those are my vessel elements. Tracheids with the pits are much longer and narrower and have the bigger holes in it. Yes?
I'm confused because on the document
Mhm.
it says Pinus australis tracheus prepared slide, and that's what that looks like.
Yeah, this is not, this is Acer rubrum. This is actually Acer rubrum. This is Pinus australis right here. And my Pinus australis, narrow ones. So here's my Pinus australis. Find a really good example of one. Some of these. That's a really good tracheid right here. That's a tracheid right here with lots of tiny openings. They're much longer and narrower. They're all going to have openings to them, and unfortunately they're going to look very similar. But the ones that have large, big openings, those large pits, tracheids with pits. Okay? So this is Pinus australis. Those are tracheids with pits. Okay? That's what they are. That's Pinus australis.
So then what are the vessel cells? Because then it says on the lab guide, it looks just like that, and it says vessel cells.
Well, they're going to be a little bit different.
Okay.
They're a little wider. Okay, so my vessel cells, I unfortunately don't have any on here that look really good. There's one right here. That is a really good vessel cell right here. They're much bigger. This is a vessel cell right here. Okay. Much wider, and it's got a little bit more of the cross hatching to it. There are going to be tiny openings in there, but the vessel elements are much wider, okay? So much, much wider there. The xylem is difficult, okay? Tracheids with pits are going to be really long and narrow with very large openings and holes. They're going to look like little flutes. Vessel elements are just going to be very wide. They're going to kind of tend to have a bit more of a cross-hatching appearance to it, okay? All right, so oh, wrong one. I got it out of here. Alright, so let's see, let's look